But now it's my distinct pleasure and honor to introduce our keynote speaker, Dr. Yaël Mossé. She's a tenured professor and physician scientist at the Children's Hospital of Philadelphia. Just a little bit of background before I start with the introduction. She did medical school at Sackler School of Medicine in Tel Aviv, and then came to the Children's Hospital of Philadelphia, where she did pediatric residency and then fellowship. And then really as a physician scientist over the next 20 years, has developed one of the most influential and comprehensive translational research programs in pediatric oncology, fundamentally changing our understanding of neuroblastoma and, more importantly, changing outcomes for children affected with neuroblastoma. Her work identified ALK as the major neuroblastoma predisposition gene and established it as the only recurrent mutated clinical actionable oncogene in neuroblastoma. This landmark discovery really resulted, as you will hear, in changes in the treatment and improvement in outcomes for patients. Through her rigorous laboratory investigation, innovative preclinical modeling, and close collaboration with national and international clinical trial networks, she has really helped make this from a target to a therapy. More recently, Yaël has pioneered entirely new approaches to harness ALK as an immunotherapeutic target. Her laboratory research has fueled the development of other approaches such as antibody-drug conjugates, CAR T-cell, bispecific antibodies, protein degraders, as we heard of earlier from Nicola, really creating other therapeutic amenities to overcome treatment resistance. And of note, I saw that Yaël is a lead investigator of a highly prestigious and competitive NCI and UK supported international Cancer Grand Challenge initiative in partnership with Cancer Research UK, with the goal to develop next generation therapies for patients with solid tumors. That is not only focused, I believe, on neuroblastoma. I would also like to highlight that Yaël has created an infrastructure that really continuously links the patient care with scientific discovery through profiling, innovative trial design, cutting-edge liquid biopsy technologies, and is really a model for what we would like to do in precision oncology. I distinctly remember two patients. This was early on in the area of precision medicine, around probably 2009. I was at the NIH and treated two of her patients on her ALK inhibitor study with crizotinib. The first was a boy from China who came to us with an ALK-mutated anaplastic large cell lymphoma. He had massive disease burden and had received a prior transplant. And it was just, for me, miraculous to see how within a week of an oral ALK inhibitor, we couldn't palpate any of the masses anymore. And the PET scan that was subsequently done showed a complete response. This patient underwent another transplant and was cured. And then the second patient was a baby. And at NIH, as you would all know, we cannot really treat very young children. We don't even have blood tubes for the little kids. But we thought, okay, this patient may actually benefit. This was a little boy from Sweden who came with his family, his brother, mom, and dad, to get treated. And this boy had a germline mutation in ALK and a tumor that was fairly rapidly growing, and he had received all prior standard therapies. So with the Yaël's help, we were able to bring this boy. It was really bad to get him to take the drug because it doesn't taste very good. But we were able to overcome all of these challenges. His tumor didn't disappear on imaging, but it stopped completely to grow. The boy went back and forth between Sweden and here, stayed on treatment for several years, and then stopped the ALK inhibitor. They recently came, like a year or 2 years ago, to visit the pediatric oncology branch. He's doing very well, has no evidence of disease. So when that happened for me, this was really gave me the hope for precision therapy that is less toxic, because there were very little toxicity. And this was, for me, the beginning of precision therapy, prior, I think, to even lower tract nip or other stories. So Yaël, thank you for all you have done and will continue to do. And please all join me in welcoming Dr. Mossé for her keynote speech.
Wow, thank you, Brigitte. I'm really humbled by the incredibly kind introduction. I want to also thank Tasha and the foundation. This is really, you can tell this is a very special meeting, a lot of friends and colleagues here. And I also want to thank Tasha and Paisley, as Brigitte said, really remind us of the fierce urgency of now, which is why we are all here and doing what we're doing and so committed to it. So with that, it's really exciting for me to be able to share more about our discovery of ALK mutations and the impact that this has had. And I'll just take a step back, and I think we all know that childhood cancer survival is a triumph of modern medicine. This was historically a lethal disease, and it's thanks to former mentors of mine and many in this room, doctors Audrey Evans, Anna Meadows, and Dan DiAngio, that there have been stunning and rapid improvements in cure rates through the latter half of the last century. And first, it took these pioneers to test new chemotherapies and radiation therapies on children with cancer. And then second, it took the success of taking a single chemotherapy drug and then combining it with other chemotherapy drugs that needed to be done in children. And third was really the formation of what are now international childhood cancer cooperative groups that have taught the world how to make progress in a relatively rare disease. And today, 4 out of 5 children with cancer are cured. But more children die each day from cancer than all of these diseases: AIDS, cystic fibrosis, heart disease, muscular dystrophy, birth defects, sickle cell anemia combined. And 2 out of 3 childhood cancer survivors have a lifelong disability, and 1 out of 4 childhood cancer survivors has a life-threatening disability. And despite the successes in the modern countries, we know that they are inverse in the low and middle-income countries, and that is simply unacceptable. So the disease we study in the laboratory is neuroblastoma. That has been, it demonstrates both clinical and biological heterogeneity and has had a clinical behavior that, as Carol can attest to, has really fascinated investigators for decades. It's quite intriguing that there can be the low-risk form of the disease shown in this baby at age 3 months, which is still widely metastatic, with all these nodules in the skin, disease in the liver, and disease in the bone marrow that without any therapy just spontaneously regresses. But the high-risk form of the disease shown in the bottom panels, where you have the birth of tumor anywhere along the sympathetic nervous system that metastasizes to the bony skeleton, is highly resistant to therapy. And still today are only curing about half of these kids. Here at a significant cost. So what is neuroblastoma? It is an embryonal cancer. It is due to the misappropriation of normal autonomic neurodevelopmental pathways. It's not a brain tumor. The median age at time of diagnosis is 17 months. It remains a very important pediatric problem. As I'll show you, about 1% to 3% of cases are, like all other human cancers, are due to, can be inherited, and this is due to mutations largely in ALK and partially in this gene FOX2B. At diagnosis, there's a paucity of mutations. We tend to only see recurrent alterations in MYC-N, ALK, ATRX, and less frequently in the RAS/MAP kinase pathway. And our current therapies are brutal, largely ineffective. And for children who suffer a relapse, today there are no known cures. So I started my fellowship in 2000 and went to the lab in 2001, and my first research project was to find the familial neuroblastoma gene. And it had been reported in 1967 by Jane Chatton, who was a pathologist at CHOP, about a family where there was a kindred who had multiple disorders as well as neuroblastoma in 4 other siblings. So that was in 1967, and then in 1972, Alfred Knudsen, a former friend and mentor of mine, and Louise Strong together mathematically modeled genetic predisposition to neuroblastoma and pheochromocytoma, and they hypothesized that just like all other human cancers, a subset of neuroblastomas would be due to a germline mutation. They postulated it would be in a tumor suppressor gene. And there's this line here that says hereditary cases are estimated to account for 20 to 25% of all neuroblastomas. And then in 1997, John Maris and Garrett Brodeur actually published the pedigree of the Jane Chatton case report from 1967. The index case is patient number 5, shown on the bottom right, and she had neuroblastoma as well as Hirschsprung disease. And then fast forward many, many years later, in 2003, a group really interested in a very rare disease arising from the neural crest known as Ondine's curse, babies born with an inability to breathe, also known as congenital central hypoventilation syndrome, identified polyalanine expansions as the cause of this disease in a gene called FOX2B. And it was only in the footnote of this figure, this table, that we noticed, John Maris and I, that 2 of these patients actually also had neuroblastoma. And so we then hypothesized that perhaps FOX2B was the neuroblastoma predisposition gene. And we went and sequenced all of the families that we had collected. We had about 30 families with DNA from multiple individuals. And it turns out that only the very first family we sequenced, which was the family I showed you twice in the prior slide, had a germline mutation in FOX2B. So this was really exciting, but this was not going to be the familial neuroblastoma predisposition. And so we took a step back, and we leveraged having now not only identified but also really banked DNA from these rare families where there is high penetrance. So you can, skipped a slide here. So we leveraged the collection of pedigrees that we had, and we also leveraged newer technologies. At that time, we did a linkage analysis using single nucleotide polymorphisms tiled across the genome. And we were able to identify a single locus suggestive of linkage. And this was on the short arm of chromosome 2, and you could see the logarithm of the odds score was pretty high, 4.23, really suggesting that here would reside a gene that was disease-causing. We had a single family that I show you here where there were informative recombinations in 2 of the siblings, one both proximally and one distally, that allowed us to really define the region of interest. And this region had 104 genes. And this was my project as we led this discovery, I then started Sanger sequencing every single one of these genes. What was interesting was that the proximal end was the MYC-N gene, and we thought for a long time that perhaps MYC-N was the disease-causing gene. But it turned out not to be, and after doing a lot of sequencing, we identified point mutations in the ALK kinase domain as the major neuroblastoma predisposition gene. And it was really thanks to this particular family. If you can see, the individual that brought this family to my attention was a young woman who shared that she had a sister who died of neuroblastoma. She had a brother with like a funny eye, and she had another sister diagnosed with neuroblastoma. And so through her efforts, we were able to collect blood and extract DNA from many of these individuals. And ultimately, really, this was a pivotal family for us to discover the ALK mutations. And it turned out that this young woman, when we then could offer genetic testing, was a carrier. And that has dramatically impacted her life. And she went on to make important decisions for herself as she got older and childbearing age. So this discovery was in 2008, and we and others showed that neuroblastoma is, in fact, has a hereditary component, and that it is autosomal dominant. and that the penetrance of these mutations is incomplete, which makes genetic counseling very complicated. And it was Karis Eng who provided a really nice perspective on the papers published simultaneously by our group and 3 others, really highlighting that ALK was likely going to become a ringleader in the neuroblastoma space. And we immediately worked with our molecular genetics lab to be able to offer Sanger sequencing to identify ALK mutations in patients. So what is the anaplastic lymphoma kinase? And I'll just tell you that when I was a postdoc in the lab, I really hesitated to sequence ALK because I sort of stuck on the fact that it was a lymphoma gene, and I couldn't really quite get my head around why this was a gene that I needed to sequence. And it is, in fact, a very important gene in lymphoma, and it was first cloned by Tom Look when he was working in Steve Morris's lab at St. Jude in 1994 in a pediatric cell line derived from a patient with anaplastic large cell lymphoma, as Brigitte was talking about just earlier. It used to be an orphan receptor, but now 2 different groups have identified the ligand. We know that inflammatory myofibroblastic tumors are kinase-driven tumors, largely due to ALK, but also other kinases. But it really wasn't until 2007 that ALK got on the map, and that's when the Japanese group identified translocations in ALK in about 3 to 5% of patients, adult patients with non-small cell lung cancer. And we were the first to identify point mutations in the kinase domain of ALK. And one of the major differences, and biologically this is really important and has been a challenge for us, is that the ALK receptor remains full length. So you have the extracellular domain, the transmembrane domain, point mutations in the kinase domain, very different from the fusion oncoproteins where you don't have the extracellular domain of ALK. We now know today that there are multiple other tumors with ALK fusions, and we know that a major mechanism of resistance to ALK inhibition therapy in adults with fusion-driven cancers is through point mutations in the kinase domain. And back then, when we set out to characterize the frequency and spectrum of ALK mutations, we did this with Sanger sequencing, and we published that for patients with high-risk neuroblastoma, 10% have an activating mutation, and independently another 4% have high-level amplification. So this provided about 14% frequency. And we today also now have shown on the bottom right, the crystal structure of both the kinase domain and of the extracellular domain to help inform our work that has been published by others. And so fast forward to today, what do we know about the frequency of ALK mutations? On the top right is the sequencing using next-generation ultra-deep sequencing of tumors from a recent COG cohort of patients treated with modern therapy. And what we now know is that amplifications are still around 3 to 4%, but in fact, 22% of patients with high-risk neuroblastoma have activating mutations in ALK. The majority are clonal, but some are subclonal. And we know that the presence of an ALK amplification still today, still with modern therapy, is predictor of very inferior outcome. The 5-year event-free survival on the top left for an ALK amplification compared to patients with wild-type disease is 25%. And for patients with point mutations, it's 33% versus 51%. So this is a therapeutic marker, a therapeutic target, and a biomarker of really inferior outcomes. So back in 2008, when we first presented this work at AACR in a late-breaking abstract, there was a scientist working at Pfizer who walked up to me and he said, I have a drug, it's called PF1066, and I'm going to drive it to your lab. And that's really what he did. And so we had sequenced all of our cell lines, and we poured this drug on the cell lines, and we started to recognize that only cell lines with ALK mutations responded, and that's on the top left. And then on the top right, when we really honed in on the ones with the different mutations that were exactly the hotspot mutations we're seeing in patients, we started to also recognize there was a differential sensitivity to this early generation ALK inhibitor. So in the Western blots, you can see that cells that have a mutation at R1275 are very sensitive to this early inhibitor. We can abrogate signaling at 33 nanomolar, but F1174 residue mutations are more intrinsically resistant. Nevertheless, this provided really the preclinical rationale to launch a phase 1 trial. And I want to highlight the date because it was August 2009, and all of the papers on ALK and neuroblastoma came out in August 2008. So this was really rapid translation, and it was in large part thanks to the commitment of Pfizer to help, to collaborate with us to bring this forward. And so we launched a phase 1 trial through the Children's Oncology Group, and we had various cohort of patients, always allowing patients to be enrolled, particularly those who had known ALK alterations. And we identified a recommended phase 2 dose that is 200% that of the adult maximum tolerated dose. And the reason for that was really because of the parallel work we were doing in the laboratory to further understand the differential sensitivity that we had seen in our cell lines based on mutation. So what this shows you first and foremost is that we did sequencing of about 2,000 tumors from patients enrolled on COG trials and found a whole spectrum of mutations. And while the vast majority are activating, their catalytic activity really differs based on their location in the protein. And so some have modest catalytic activity. Others, for example, those in the phenylalanine core, are highly active mutations. And then there are actually some that are not. Those that are located in the C lobe don't appear to have a functional consequence. And so this was important in understanding potentially what patients with what mutations would benefit from an ALK inhibition strategy. And then we had insights from the crystal structure of the kinase domain of ALK that had been solved and looked very similar to that of EGFR. And we learned a lot. We had already knew a lot in the field about EGFR, and I had the fortunate opportunity to work with Mark Lemon, who was the chair of biochemistry at Penn at the time. And what we were able to understand is that mutations, that this residue F1174 is in close proximity to this really critical DFG motif, and that is responsible for the transfer of magnesium ions, which is important for ATP binding. And when this residue is mutated, there's enhanced ATP binding affinity. Importantly, what we were able to elucidate from the structure is that crizotinib doesn't bind anywhere near the DFG motif. And so even in the presence of a mutation at this residue, it should still allow crizotinib to bind. And so this provided for our laboratory a structural hypothesis that if we could push the dose, we could, and infer that crizotinib would still bind, we could potentially overcome this preferential ATP binding affinity in the presence of a de novo resistance mutation at this residue. And as Brigitte alluded to, it was a lot of fun to run this trial. And what we learned is that crizotinib is really active against childhood cancers that have an ALK fusion. So on the left, I show you the data for the 26 patients we enrolled with ALK fusion positive anaplastic large cell lymphoma. We saw an objective response rate of 90%, and these phase 2 data led to FDA approval in January of 2021. You might wonder why some of these patients stayed on for such a short time, and it's really because it was so unexpected, and these patients had dramatic complete responses. And just as Brigitte said, then the treating physicians took them off study and took them to what they thought was definitive therapy with either auto or allo transplants. And then for the rare patients who have these inflammatory myofibroblastic tumors, we saw also a really impressive response rate of 86%. And we were able to, again, with Pfizer and the support of the FDA, just with 14 patients get FDA approval. But ALK-positive neuroblastoma, as we predicted from the preclinical work, was a different story, and the objective response rate was only 15%. However, and again, thank Brigitte for mentioning this little boy who came from Sweden. We treated 2 children with germline mutations. As I mentioned, this was the original discovery of ALK, was these patients who have germline mutations. This little girl, she came to see us. She was born with neuroblastoma, diagnosed at around 4 months of age. She had bilateral adrenal tumors and a tumor in her chest, so she really had 3 separate tumors. When we met her, she had had multiple rounds of chemotherapy, 4 major surgeries, and despite the lack of any family history, because of the bilateral adrenal tumors and the suspected genetic predisposition, we worked with our molecular lab, had developed the genetic testing tools, and we found that she had a germline ALK mutation. And rather than recommending intensified high-risk therapy, we recommended as best as we could with the equipoise that's needed when you participate in these trials and are also doing the preclinical work, enrollment on the phase 1 trial of crizotinib. And she had a complete response after just 3 cycles. The radiologist called to tell me actually that the surgeon had done a great job. And that was not the case. It was this oral medication that does taste absolutely awful that had done an amazing job. And we kept her on therapy for 5 years. And she's going to be 17 tomorrow. It's really amazing. And so when we went to test her parents, it turned out that mom was the germline carrier. So there was not de novo in the little girl Edie, but her mom was a silent carrier. And when mom, after genetic counseling, decided to became pregnant again, when she was 30 weeks of age, 30 weeks pregnant, she started complaining of back pain. And lo and behold, an ultrasound showed the growth of bilateral adrenal tumors. And you can see the MRI here. It's really quite amazing. You can see the fetus, and then you can see the bilateral adrenal tumors. And so they moved to Philadelphia. We prematurely delivered the little boy, Kinsey, who I failed to mention, we had done prenatal testing through chorionic villi sampling and were able to share with the family that he had not inherited the mutant allele. So he was not going to be at increased risk for developing neuroblastoma. But we now treat mother and daughter, treated mom for 3 years, and they come to see us, and we treat them like patients with cancer predisposition syndrome and do very close monitoring and surveillance with MRI and ctDNA. So this taught us that this is really where the science came to life, where we started to recognize that some patients really are going to benefit substantially. And since we really only had crizotinib at that time, we did some work in our preclinical models. This is one derived actually from a former patient of ours whose tumor had an ALK mutation that was also resistant to crizotinib, but we could restore sensitivity when combining with chemotherapy. And this provided the preclinical rationale to move crizotinib up front in the pivotal phase 3 trial opened by the Children's Oncology Group in the spring of 2018, where at that time we were still doing Sanger sequencing, and any patient identified with an activating ALK mutation, as we had defined it in the lab, would be non-randomly assigned to Arm E and get crizotinib throughout their therapy. In parallel, we were working very hard in the laboratory to identify better, more potent ALK inhibitors, and ultimately this led us to the observation that lorlatinib, which also happens to be made by Pfizer, but we had looked at every other ALK inhibitor that had been made by different companies, and that was about 10 at that time. And lorlatinib, you can see, has a very different chemical structure. It's a macrocycle, so it's a ring. And I show you on the left panel that its activity in red against patient-derived xenografts that have mutations that are resistant to crizotinib in blue. And so in red, we saw complete and sustained responses, and we had not really ever seen this before. And in the bottom right is a model that is sensitive to crizotinib in blue. As we had shown, this is the mutation that responds to crizotinib, but if you keep treating, the tumors become resistant, whereas with lorlatinib, we were able to maintain tumor regressions. And so we were really excited about this, and we translated this to a phase 1 trial in the New Approaches to Neuroblastoma Therapy, the NANT phase 1 consortium, where we, again, in close collaboration with Pfizer, opened 3 different cohorts: young children, adolescents, and adults, because for reasons we still don't understand, we see a higher frequency of ALK mutations in older patients, and then a cohort of combining with chemotherapy just to show the tolerability. And this really was the first time that we also integrated serial profiling of plasma to look at what we could learn from circulating tumor DNA. And we showed a couple of years ago, we had always said in neuroblastoma, wouldn't it be nice if one day we can actually have some waterfall plots that actually show response in these patients? They're very heavily pretreated. And we did, in fact, it was validation of the preclinical work that for a subset of patients, we were seeing really robust and objective responses. And so this led to a major amendment of an ongoing phase 3 trial that I mentioned, the ANBL 1531, where midway we really argued that patients deserved this therapy now rather than waiting for the next trial. And so we switched out crizotinib for lorlatinib, and we actually also brought next-generation sequencing forward so that we could identify patients even with more subclonal ALK mutations who we think can benefit from this therapy. And today, even though ANBL 1531, all the other arms have closed, Arm E is still open. So any patient with high-risk neuroblastoma, in my opinion, should receive therapy on Arm E of 1531 to see if we can really improve survival. And we are learning in real time the response and mechanisms of sensitivity and resistance by really leveraging the technology of liquid biopsies. We published 2 really fun papers about this, and this was using the Foundation Medicine platform, but that is developed specifically for adults, and it has 400 genes, and a lot of those genes are not relevant, and then you use up your, you have less ability to really do a deep dive and ultra-deep sequencing in the genes that are relevant for neuroblastoma. And so we took on in the laboratory, in my lab with Esther Burko, who's a very talented now physician scientist and living in Israel, making a custom panel for children with neuroblastoma with the genes that we think are most relevant. We translated this to our molecular diagnostic lab, so this is now CAP-CLIA approved, can be accessed from anywhere around the world. We make it very easy. This is an example of a report on the top right of the information that we provide with relatively rapid turnaround. It's a custom pipeline where we do a lot of manual review, and it's really quite deep. And so we are able to detect, really use this for minimal residual disease monitoring. And we're now pushing the boundaries of ctDNA, as many people are, to develop a mutation agnostic assay. And so from serial ctDNA profiling on the lorlatinib phase 1 trial, we learned that the major mechanism of resistance is on target. So it's again in ALK. ALK is really like the major vulnerability. And so on the left, I show you in green the parental mutations that are there. And then upon treatment with lorlatinib, we see in about 40% of patients who are treated largely at subtherapeutic doses that I think breed resistance, the acquisition of a second mutation on the same allele, so in cis, also in the kinase domain of ALK. And in parallel, so on the left is the clinical data. On the top right, we took a neuroblastoma cell line with a ALK mutation, and we used CRISPR to engineer in cis these new mutations to prove that they are, in fact, the mechanism of resistance. And you can see that the IC50s shift to the left, or rather shift to the right upon the acquisition of a mutation in cis. And we were modeling resistance in parallel to the clinical trial. So on the bottom right is a patient-derived xenograft that has a mutation in ALK at residue F1174. And we took the mice and we treated them for years with lorlatinib. And as the tumor started to slowly grow off, we would perform surgery, reimplant them, and then they started to regrow much faster. And we indeed found a compound mutation, as we call them, in ALK as well as the major mechanism. And so we are now really trying to think hard and outside of the box for how to overcome this resistance or prevent resistance. And we are moving beyond inhibitors and moving to the space of targeted protein degradation, as Nicola showed. And we are very fortunate I'm co-leading a Cancer Grand Challenges team with Martin Eilers, where we have brought together international expertise in this space, along with an industry partner, Therapeutics. They're in the Bay Area and San Francisco, and they're really at the forefront of developing degraders for cancer and autoimmune diseases and inflammatory disorders. And we're aiming to develop degraders for 5 childhood oncoproteins, one of them being ALK, where we aim to prevent or overcome resistance and also address the non-catalytic functions of ALK. we are targeting a ubiquitously found oncofusion protein in the rare lethal liver cancer fibrolamellar hepatocellular carcinoma, driven almost exclusively by this DNA JB1 PRCACA fusion. And then, like Jeff and I were talking about earlier, now the targeted protein degradation space gives us an opportunity to also go after what were previously undruggable targets. And this is a recent picture of an annual retreat that we had in Würzburg, and the top right picture is meant to highlight how important advocacy is in our work. And the advocates are a real part of all of the scientific development. And I don't need to really at all go through this slide. Nikki did a great job of introducing what are PROTACs, how they hijack the cell's natural ubiquitin proteasome system to eliminate disease-causing proteins. And there are two different classes of PROTACs. There are the heterobifunctional degraders, classically called PROTACs, and then the molecular glues. And we're exploiting both modalities for the childhood oncoproteins that I just mentioned. And this is just an example of the workflow with our industry partner. It's highly collaborative. They have a proprietary DNA-encoded library that allows us to screen for ligands. They provide the upfront discovery work through their DNA-encoded library discovery work, their ligase enablement, all of their really high-throughput chemistry. And then the partners in academia provide the expertise in the disease biology, the models, and the translation of the work, and really the optimization and testing of all these compounds to get them to the patients. So I'll give you just an update on how we're doing with ALK. This has been the lead program because we did not have to use a DNA-encoded library screen due to all of the existing chemical matter, all the small molecules already being developed and already in patients. That can serve as your hook. And so we've done a lot of, it's been now about 2.5 years that we've been working on this project. We've identified on the top left lead compounds that have similar potency to lorlatinib in vitro, that have differential sensitivity against cell lines that have both just a single mutation as well as those with compound mutations. On the top right, I show you that at very low nanomolar concentration, 10 nanomolar, 33 nanomolar, we start to see near complete degradation of ALK, which exists in two different forms, in cell lines that have compound mutations, so very resistant. In vivo, we see degradation in the Western blot, and then we've taken a number of our lead compounds and started to look at them in vivo. And this is the first in vivo study ever performed, where we only had enough drug to treat for 3 weeks, and you can see that some of them, of the degraders, are better than others. So this has really been an exciting program for us. And here is really what I show you is the iterative nature of degrader optimization. So we started in 2024, on the x-axis you can see time, and on the y-axis is potency. So we started with some not very potent molecules, and over time we've really optimized these to become more potent. We've demonstrated in vivo degradation, and we now have about 400 new degraders where we have picomolar potency, and importantly, we're also starting to dial out some of the vulnerabilities that we don't want. We know one of those is inhibition or degradation of TrkB, so we're able to dial out liabilities. So this has been really exciting. And in parallel to targeting, using multiple modalities to really target the point mutations in the kinase domain, again, Brigitte alluded to that we've been really interested in the fact that the receptor is full length and retains expression of the extracellular domain. And as Brian first mentioned in his talk, ALK is a true oncofetal protein. Its expression is limited to the developing nervous system before birth, after which it is entirely turned off. And that is shown on the top left from the EVO-DEVO database. On the bottom is RNA expression data of neuroblastoma compared to a multitude of normal tissues. And you can see that it's really only minimally expressed in the immune-privileged organs, such as the pituitary and the testes. And when we look at a human tumor microarray of neuroblastoma tumors, we see differential expression, but brown, all of that is ALK, and it's not expressed at the protein level on any normal tissues. And interestingly, ALK is also expressed across other histologies. So it's actually been well documented that it's expressed in the vast majority of fusion-positive rhabdomyosarcomas, as well as a host of other histologies. So we took the approach that we wanted to develop a way to target cell surface ALK, and the first approach we took was through the development of an antibody drug conjugate, which we actually made in academia, as shown on the bottom left. We were leveraging a DNA alkylating agent as the payload. And what we've shown and just recently published is that across neuroblastoma models that have all different genomics and are highly, highly therapy resistant, some of these have ALK mutations, some of these don't, some have MYC amplifications, some have P53 mutations, but we really saw exquisite sensitivity, and it was related to the amount of ALK on the cell surface. So really the only model that expresses very low ALK is the model SKNAS, and even there we still saw, because of the really potent internalization of our antibody, some activity. And so we now actually have an industry partner for this work, and we're working with a CRO that has expertise in this space, and that's what I show you on the bottom right. This is actually going to be our GMP grade ADC that we plan to get to the clinic for children who have ALK expressing tumors, including neuroblastoma and fusion-positive rhabdomyosarcoma. And in parallel, to continue to leverage the cell surface expression of ALK, we are developing CAR T-cell therapies. We've been fortunate enough to be able to pan for binders. We have multiple different binders. That's created actually a really complex workflow for us to really screen these scFvs in different orientations with different co-stimulatory domains. But we have now some proof of activity in our in vivo models. The one I show you in the middle, NB1, has very high ALK on the cell surface, and there we see that 2 of our constructs show complete and sustained regressions. The challenge we're really facing, or one, I should say, there are so many challenges in the CAR T space for solid tumors, but is the right model where there's less ALK on the cell surface. So we're really trying to tune with Evan Weber's lab these CAR T cells to recognize ALK even when it's present at lower densities. And so I'll leave you with this last slide, which is really a cartoon of where my vision is of building an ALK toolkit to really exploit all of ALK biology and to bring these therapies up front. That's what we have to do. That's where we're going to make the biggest difference to prevent relapse rather than treat relapse. And so, my vision is that wild-type ALK is expressed on most patient tumors, so we could really come in with an ALK ADC early on to debulk the disease. That's what these precision chemotherapies are able to do. We will learn that at least a quarter of these patients will have a mutation. And so we would like to bring the degraders forward that we think are going to be less prone to resistance than the inhibitors. And then for patients who hopefully with the improved ctDNA surveillance that we can do, where we can detect minimal residual disease, we envision coming in with a CAR T therapy to provide long-term immune monitoring and immune surveillance to prevent relapse. With that, I want to thank the patients who inspire all of us, and our, said at the beginning, the quote, "The fierce urgency of now comes from the father of that little boy on the top left, Gavin Lindbergh." It's his dad, and this was Evan, one of the first patients with an ALK mutation, not successfully, and learned a lot of lessons from him. And want to thank my lab, John Maris's lab, and the many funding sources that I'm very privileged to have to do this work. Thank you very much for this. Thanks.
I'm intrigued to see that you have been able to get industry partners, and I really wonder would love to hear more. Is this because it's also relevant in adult cancers, or how did you make this happen that I guess they are partners to potentially commercialize this? Is that what the goal is?
You know, I will start with one word, which is perseverance. And I think we all know that that's really important. What we do. You know, when we first made this observation, we expanded this, we looked for ALK expression in other histologies because that's what we thought we would need. And I went knocking on the door of so many industry and biotech companies, and they were not interested despite the potential application to adult histologies. And ultimately, it's really through, again, persistence, perseverance of finding a partner who is committed to actually translating this to pediatrics first. You know, my fear about industry partners, when you have an adult indication, they'll go for the adult indication and peds will follow maybe many years later. It really has been just, I think this is going to be such a incredibly potent, and you know, the class of drugs too is very attractive. Now the ADCs are showing promise. So, you know, I again, just perseverance if you really believe in something.
Or rather the other mutations that may be present in those low-expressing cells, for instance. Like, does that low-expressing cell, I think if I'm not mistaken, right, you referenced earlier RAS-MAP kinase mutations, some will have an F1 loss or a RAS mutation. FGFR, I think, has also been seen. So if you're low ALK, is it something else, and is that what's driving the differential sensitivity to an ALK ADC?
So thank you for that question. And one of the most compelling things I think about an ADC is that you can get pretty low because especially if you have a binder that internalizes and then, you know, traffics to the right place, and the bystander effect of ADCs allows even cells that are null for your target to be, you know, to be recruited and killed by the poison that's accessible to the cell. And I think the challenge is that in the low cells, oftentimes, those are very rare, actually. So most neuroblastoma cells have pretty modest levels. I would say that the challenge for the low levels of ALK comes more when leveraging the cell therapies. So I think ADCs are actually going to be broadly relevant because of their bystander effect, regardless of underlying mutation. Even in the presence of P53, have, you know, a payload that can overcome that. I think it's going to be much harder for CAR T cell therapy to overcome this challenge of cell surface, you know, density, heterogeneity.
Tumor gene with variable penetrance, and so why the variable penetrance? Is this a two-hit theory? Are there other genes that you're looking at that are combining with this gene?
Yeah, thank you so much for that question. It's something that's, ever since our initial discovery, really, it's a question that we have to answer. And we've not found two hits. We don't know what the second hit is. And so that's of interest, that it's a heterozygous mutation in the patients, you know, who have it in the germline, and we don't see amplification of the other allele. And so we don't know what the second hit is, so that's first. And then secondly, due to the incomplete penetrance and the diversity of phenotypes, even when it is penetrant, that you can have, you know, in the same family, a patient with low-risk disease and a patient with high-risk disease, there are absolutely going to be other genes cooperating with ALK. And so we're leveraging the families that we have to answer exactly this.
Sorry. I know you briefly mentioned the use of CRISPR, and I was kind of curious about the case with the mom and the daughter and how you found like a genetic aspect in that case. I wonder if it were found any earlier, if CRISPR could be used, and especially with like all the progress as in the last couple of years.
Yes, it's, and it gets back to actually the incomplete penetrance of this disease, which makes the genetic counseling and the potential role of prevention very complicated, because the penetrance is about 60, 65%. That means that even if you are a carrier of a mutation in this gene, there's about a 35% chance that even if you pass on the mutant allele, your child will never have neuroblastoma. It becomes complicated from an ethical standpoint.
Really extremely fast and impressive, I think. Could you still do this today? Or I feel from my perspective, it takes so long to get clinical trials up and running.
And I think, Brigitte, thank you for that comment. I think we have to be, even more nimble. I think we need to not only get to a clinical trial as fast, but we need to get up front. There's too much time between discovery, phase 1, but then phase 1 to phase 3 just takes too long. I think we need to be more thoughtful about doing small studies and then moving these drugs up front when there is rationale to do so.